All right, good morning. That's a lot of people. Thank you all for coming. I hope that after my talk, everyone would be convinced that microsatellites are actually very important. So I'm just going to start with a short introduction on the fact that we know that 70% of, or maybe even more than, of the nucleotides are thought to be transcribed. Many studies have shown that. And most of us just usually study the coding transcription. And I want to shed a light on more of the non-coding transcription and mostly the one that happens at repeats. So as our assemblies of the genomes have become better and better, the part of the unknown regions are starting to come as also repeats, especially in the last assembly from the T2T consortium, which has been shown to be more accurate. And in that assembly, right, we have a large increase of number of repeats that have been shown to be there. And for me, that's quite interesting to study those kind of novel regions of the genome. And today I'm going to mostly focus on short-term repeats. So just a few key brief definitions for those who do not know what short-term repeats are. Those are sequences that are made of short elements between 1 of 6 base pairs that are repeated. And just some key terminologies. We usually call the STR class the element that is being repeated. So here we have, for instance, a TSTR and ACSTR. And we also call the length the number of the repetitions, and the flanking sequences the sequence that are on both sides of the repeat sequence. So those repeats are quite difficult to study because by their nature, they are difficult to map in sequence. But the key technologies have allowed us to do that in a better way, such as long reads and better short read sequencing technologies. It has been shown also with quite many studies that those repeats, they can impact transcription in a functionally relevant way in things like binding or spacing or splicing or even nucleosome packaging and histone modifications. And there are many different functions that have been associated with short-term repeats. And so it is, I think, quite important to try to understand what are all the different kind of mechanisms taking place there. And particularly, I want to speak about one example that I think is interesting, is in for a particular gene, it has been shown that the microsatellite was acting as a regulator of the gene, where this WSFA1 gene was bounding, like nearby that gene was a GGAA-STR was bounding, and if it was unbound, then we will have no transcription and also no transcription of the gene. And what the people in that study did in 2018 is that they took a tumor where the STR has been silenced and transplanted that tumor into a mouse. And they have been showing that the growth of that tumor has been impaired when the microsatellite was silenced. So there are probably other instances where microsatellites can have clinical relevance when they are being acting as regulators. So in the previous studies, we have also had better ways to look at the genome. For instance, in the publication of the Phantom Mammalian project in 2014, where the Phantom consortium used the cage technology to map a very precise picture of the transcription start sites along the genome. And during that, in those libraries, they were also able to show some peaks, like some places where they have been identified as peaks. And we have also shown that a large portion of those peaks were initiated at STRs. So this is also proof that STRs are initiating transcription, and other similar mechanism can happen where the transcription of STRs can have an impact of the transcription of other genes. So once we took our hands on that data, we decided to create a more continuous signal at the STRs, because a peak is a binary signal, and that's something that is has its properties, but if we want to study a more continuous signal, that's not ideal, right? So we decided to build what is called a tag count signal, where we average the cage peaks over a large amount of libraries. So in the cage, in human, there's about 1,000 libraries in cage, and we also average that over the length of the repeats, so that every single repeat can be associated with a single measure of transcription. And then when we published that result, we also noticed that the different classes of the STRs also tend to have different levels of expressions. So then maybe different classes of STRs have also maybe different functionalities or different impact on the transcription of genes, for instance. And something else that was also interesting is that when we look at clinical variant databases such as ClinVar that are replicating variants that have a clinical significance, the repeats that have ClinVar variants tend to be more expressed than the repeats that do not have ClinVar variants. So that's also something that's quite interesting, as shown on the plot on the top. And then in ClinVar, the variants are also classified based on their pathogenicity. And we've also shown that when the variants are classified as benign and take part in 2 STRs, then those STRs tend to be less expressed than if they would have a pathogenic variant, as shown on the plot on the bottom. So there seems to be a link between the clinical relevance and the transcription of microsatellites. So once we have all that information, something that we can do to try to better understand is to model the transcription at short-term repeats, or model the transcription of any other type of sequence. And for that, we developed a interpretable architecture that was specifically designed to do this. So this is a deep learning model that is not very deep, so it can be easily interpreted and interpretable. And so how it works is just like any other model. We take a DNA sequence and we transform it into a one-hot encoded metric. That's very classic. And then we pass that into that very simple architecture where we have a model that is composed of modules. That's why we call it a modular neural network. And a module is a very simple unit where we have a single 1D convolution network or convolution filter. And then after that convolution filter, we have just a simple multiple linear regression. So that we're going to take all the outputs at each position of the sequence from that filter, and we're going to string that into one value. So each module is outputting one value. And then when we have multiple modules, we are going to do another regression between all of the outputs of all the different modules. So something that is quite important to note is that this model is not linear because it has some value activations. And it's by design interpretable, because by just looking at the 1D convolution filter, we can know the motif that the model is looking at. And by looking at the first multiple linear regression, that is called number 2 here on the plot, we can see where is the model looking at that motif. And then finally, by looking at the regression between the modules, we can know which modules or which motif contributes to the transcription in what ways, in a positive way or a negative way. Another innovation with that model is the way we train it. So we use a method so that the model is architecture agnostic. The architecture of the model, the specific hyperparameters of the model are going to be found by the model itself as it is training. So how do we do that? We just have our training and test set, and we use a library called RegTune that is going to find the best parameters of a module using a genetic algorithm. So we are just going to train many different types of modules with different filter sizes and different learning rates on the same data set. And we are going to iteratively promote the modules that have a better performance until we reach some epochs, some iteration of that algorithm that we think that we have a good module. So once we have a good module, we are going to freeze the weights of that module. So the model has run a rough estimate of the dataset, and we are going to restart the process by adding a new module to the one that has been frozen, so that the model can have a way to refine that prediction. And hopefully this will improve performance. And once we have reached a few epochs of that process where we do not anymore improve the performance, then we choose to stop the process and we think that we have a final model. In this way of training, we ensure that the model is learning motifs that are orthogonal to each other, because the motif cannot cooperate. And the only way to improve performance is to find a motif that is different from the one that had been learned before. So in terms of performance, those models have a much fewer parameters, so they are much easier to interpret. And they are close to the original CNN model that we published that has much more parameters. So once we have model transcription of the STRs, what can we do to have a better picture of the functional impact of the transcription? So I want to put emphasis on a study that was done by Guillemard et al. In 2016, where they found a way to assess the importance of the length of the repeat on the nearby gene expression. So they take a population. In their analysis, they took the GTEC population, and for each individual, they just measure the length of a STR within a window. This is for 100 kb, and they try to correlate the length of the STR with the expression of the nearby gene. So in this plot, this mock plot, this is not real data. Every point is an individual so we can see the length of the STR and the gene expression. And if we have a positive or negative correlation with significant, we then think that there is association, right? In our case, we would like to use transcription instead of length because it has other properties, and it has been shown that those two biological meaning that data sets are orthogonal. But most data sets actually do not measure transcription of STRs because these are difficult to map. So instead of using the transcription, instead of using the length or the transcription that we do not have, we use an imputed version. So in this case, it is the modular neural network score. So how do we do that? We use a in-house developed pipeline where every individual has a variant call, so we know which are the variants in the STRs, and we impute the variants in silico, and we apply those models to this modified sequence. And then, so for each individual at each STR, we compute the score, and we try to, as before, just try to find a correlation between the score at the STRs and the gene expression that is measured in GTEC. So if we try to do that, we find about 14,000 significant associations, where we have 115,000 STR gene pairs that are tested. And we also find enrichments between our findings and the previously published datasets. So for instance, EQTLs or splicing QTLs, and also the ESTRs that are the previous published datasets by Guillemard et al. And once we have those correlations, and we have the significant genes, STRs that are regulating genes, what we can do is that we use and leverage the modular neural networks interpretability properties. So we can do, for example, this very simple experiment where we have our filter module that is a typical convolution filter that can act as a PWM. And we can just scan the filter over our important STR that we have associated, and we can look for hits, so where we have a positive convolution score. And then we take all those short hits that are sequences, and from it we can make a PFM, position frequency matrix. This is the result, for example, of one of those analyses. This is still ongoing work, where we have on the top plot the position of the hits. So we can see that the hits mostly happen at the short random repeat end. And then on the bottom, we have the PFM motif that has been found by that analysis. And it's has been associated with the CA initial element. So an element that is usually associated with transcription. So that's the analysis that we're doing now. Is really still ongoing work. So from that, I just want to give you some key takeaways. So the first one is short random repeats do initiate transcription. That transcription can be modeled by a low complexity interpretable deep learning model. And I think that building those models is actually quite interesting. And then sometimes we do not need many parameters. The transcription initiated short random repeats is functionally relevant. And interpretability of the low complexity DL models, they can help us to uncover relevant motifs without using third-party integration tools. So that being said, I want to thank all of you for coming and for your attention. I also want to really thank the organizers for having me there, and also all the people that collaborated on the project, and Sanofi for sponsoring me during the PhD.
When you show your motifs, the motifs themselves don't look like STR motifs. So could you just explain the disconnection there between your definition of STR and
those motifs,
the motifs that actually show up, which are actually higher complexity than STR?
Yeah, of course. So what we have found is that most of the important stuff happens in the flanking regions of the STR, and the model takes a region that is a bit longer than the STR. So, so for instance, here you see the PFM motif has a bunch of T's, because this is a T STR, but the important stuff happened right after the STR or right before.
How do you know the actual STR is relevant?
Because we use a, so you want to know how do we
know the actual STR? Like, what if you remove it and run the model again? If the model is not paying attention to it, what does the STR do?
So our model is length agnostic and also STR agnostic, because the way we normalize the Y. So that's why we also find very different results from the GIMAC studies, because they use the length, which is also very typical. But we use more the flanking sequences, and we have actually tried to remove the STR itself. And when we do that, we still see quite a bit of performance. And that's also one of the key findings of the study, is that what is important is the thing that are just right below, before and after. And also something that is quite interesting is that when we try to classify the STR based only on the flanking sequences, we also find which type of STR it is. So you can make a classification. So then the imputation, the STR depends on the flanking regions, and also the length also depends on the flanking regions. So it's a whole thing.
So thank you for your talk. Always happy to hear about STRs. I noticed in some of your figures you make distinctions between classes of poly A and poly T STRs.
Yeah.
Whereas they're in double-stranded DNA, you have of course both.
Yeah.
Is this an important distinction to make in your experience?
Yes. So we train a model per class of STR, and obviously if you have a STR, a T and a plus strand, you have an A and a minus strand. So the analysis is actually stranded. So, for example, we are going to consider if we have a T on the plus strand, we pass that into the T model, and if we have the A in the minus strand, we pass that in the A model. And so we do make the distinctions between the T and the A. And actually, if you would like to swap the models, the T models and the A model, they would perform much worse than the opposite class.
Okay, so you claim that your motifs and kernel that you're going to find are not going to be repetitive or redundant, and they're going to be orthogonal one to the other. I wonder if you have any, and this is a common thing with CNNs, I wonder if you have a theoretical a proof or guarantee or any empirical results that support the claim?
Yeah. So I'm not showing that here, but we have simulated data to try to prove that. And my explanation is that if you don't use orthogonal information, you don't improve performance because you're using just the same information as before. And when we use genetic algorithm to add modules, we just select the most fit candidates for the next step. In this case, we ensure that they use orthogonal information. Otherwise, if you use the same information, you just don't improve performance.